Hi, my name's Skylar, and today we'll be talking about DNA mutations, more specifically point mutations. Here you see the original DNA sequence, which is transcribed into the RNA sequence shown below. The template strand is read 3' to 5', and RNA is made in the 5' to 3' direction. First, we must identify the start and stop codons to determine the reading frame. Here we see AUG, the start codon, and 4 subsequent codons and their resulting amino acids highlighted in the chart. The stop codon is UGA and is underlined. Here is an analogy using train cars to help better understand the translation process. The codons represent the train cars, while the cargo inside represents what amino acid the codons code for. The amino acid chain is the specific protein produced and is represented by the entire train. Here is an example of a Missence point mutation. A single base pair is changed, resulting in a change to the resulting amino acid. Originally, the ACU codon transcribed in the RNA from ACT resulted in threonine. Now, its AAU resulting in asparagine. This is a transversion, as cytosine, the purine, is changed to adenine, a pyrimidine. In terms of the train analogy, the train car or codon is switched out for another train car with different cargo or the amino acid. Here is an example of a nonsense point mutation. A single base is changed, resulting in the original codon being changed into a stop codon. This is a transversion, as cytosine, the purine, is changed to adenine, a pyrimidine. Here, the train car that represented UGC carrying the cargo cytosine is now a stop codon, so no train car is needed as there is no cargo to carry. Here is an example of a silent point mutation. A single base pair is changed, resulting in no change to the amino acid. As you can see in the chart, AUU codes for the same amino acid isoleucine that AUC codes for. This is a transition, as thymine is changed to cytosine, both pyrimidines. In terms of the train analogy, the train car or the codon is switched out for another train car, but holds the same cargo or codes for the same amino acid. Here is an example of a read-through point mutation. A single base is changed in the stop codon UGA, changing it to UGG, a codon that codes for the amino acid tryptophan. This is a transition, as adenine is changed to guanine, both purines. In terms of the train analogy, another train car is added on the back carrying cargo, or the codon UGG, which codes for tryptophan, is added.